Hey everybody. So today's video is going to be giving you an introduction to the photosynthesis simulation challenge questions, which really are just an introduction to how to run the photosynthesis simulation, which you're going to be using towards the end of this week, beginning of the next week, to identify a variable that you'd like to manipulate and in order to collect some data and make a claim about photosynthesis, or to verify a prediction about how photosynthesis would behave under various environmental conditions. So you'll have opened this document. You should have read, it asks you to open a link, okay, and then identify some of the variables. Please take a moment to familiarize yourself with that. And then there's going to be a challenge question. The first challenge question is to run an experiment with all the values at their starting position. You're going to be noticing that ATP and NADH concentrations increase and decrease at a regular rate over time. And you're going to be asked to explain the relationship between those. Okay, so whenever you're asked to explain, what you're really trying to do is to figure out why do they behave that way. You're not just describing the relationship, you're explaining why that relationship happens the way that it does. So here's the simulation, and move my screen just a little bit so it's a little bit more in your field of view. You'll notice you have two graphs here, okay? Across the top are tabs, which allows me to look at different dependent variables isolated from the others. But then at the top, I'll see them all graphed together. So this is the starting position. Everything's in its basic position. If you ever wanted to return to the start position, you can simply click reset down here, right? So I can change a bunch of values and I can be messing around and doing stuff. And then I want to reset, boom, everything's back at the start. But if I run this, you're going to see a graph at the top with 4 different lines on it: ATP, NADPH, glucose, and oxygen. And then down here, I can isolate any one of those. So I could look just at ATP or just at glucose and so on. For now, I'm going to have it set just at glucose because that's my target molecule. The challenge question says that ATP and NADH concentrations increase and decrease at a regular rate, and I want you to explain the relationship between those increases and decreases and the increase in photosynthesis. Okay, or I'm sorry, in increase in photosynthesis and the production of glucose, which I've isolated down here. Right, so I'm looking at the blue and the green lines up here going up and down in a regular pattern. Or I can pull them out and look at them individually. Notice some stuff about that. Notice some stuff about the NADPH. And then I can look at the glucose on its own. Right. So that's all you have to do for the first challenge question, is to try to connect what we're seeing here with the process of photosynthesis. If you can't do that, there's a little on the challenge question. There's a way out if you can't do it. Don't go there first. I really would like you to try to do this. You can think of this a bit like a FRQ style question on an AP test. This would be exactly the kind of thing they would like to see you be able to do. So you should challenge yourself. Go to your notes. Look at what you know about the process or have information on about the process of photosynthesis. Compare it to how the data is being presented here. See if you can generate an answer. And then if you really feel stuck or you just want to see if you're getting it right, go to the video link there. Second challenge question. All right, so our reset values, we have everything reset back to the start, and then we're going to toggle some variables in a specific way. At the top, we're going to toggle the light spectrum, or the toggle, to light spectrum, and then we're going to select the live button at the bottom and change to the glucose tab. Okay, so if I go back here, what that means is I'm going to toggle that. I'm going to click on the glucose tab if I'm not already there. And then I'm going to hit live. Actually, I'm going to reset before I do that to get rid of what I had there. Glucose tab, hit live. Okay, and now the graph is going to look a little bit different, but you'll also notice this light spectrum here is different. So this box here shows me the kind of light that's being shined through the chloroplasts. And so in the specific light spectrum, we start off at 680 nanometers. That's the wavelength of the light. So it's really only red light shining through. And so contrary to maybe what you might imagine, plants don't use all of the light from the sunlight. They use some of the light from the sunlight, and we can break sunlight up into different wavelengths. This is what happens in a rainbow. If you've ever seen light shining through a prism, you see different colored lights. Those are each of the lights that are contained in the white light that was coming into the prism. The prism breaks them apart, and what comes out the other side are the different wavelengths of light that were actually contained in that white light. Well, chloroplasts don't use all the light that's in the white light. It all shines on them, but some of it they reflect, the green light. That's why they look green. And some of it they absorb, things in other spectrums. So that's what's been manipulated in this particular challenge question, right? So then if I compare glucose production in this situation to the ideal settings, I'll see that glucose is only produced one time, just after 30 minutes. And so you see here in the original scenario, if I reset everything and I run it live, there were 2 glucose productions. But if I change the light spectrum here, I only get 1 glucose production. Okay, so the second challenge question is going to introduce you to what the photosystems use in terms of light wavelength, a little bit what I told you before. And then I want you to play around with the other variables, except for the wavelength of light, and identify one way to change the environment that will produce glucose at a faster rate. Okay, and I give you the hint, like, think through this. Look at the different variables. What are some of them, right? Like, I can change the light in the dark. I can change the distance from the light, so there's less intense light coming in. I can change the pH of the stroma. I can add NADPH. I can add more or less carbon dioxide. I can increase or decrease temperature. I can add atrazine. Atrazine is an herbicide. It kills plants, and it does that specifically by affecting photosystem 2, one of the photosystems that is involved in the first stage of photosynthesis. So mess around with those, slide them back and forth and see what happens, and see how it changes the glucose production, and see if you can change the rate. So the rate here is about 1 glucose molecule every 31 minutes or so. So can you cause this to produce glucose at a faster rate by manipulating some of these variables? The only way you can't change, light. We're leaving it at 680 nanometers. Once you've identified one that will change that, you're going to identify the variable, the manipulated variable that increased the rate, and then explain why. And again, go back to the process of photosynthesis to do so. All right. If you get stuck on challenge question number 2, challenge question number 1, the answer there should give you some sense of how to explain challenge question number 2. All right. All right. Good luck, everybody.